Hi, everyone. Today we are talking about Chapter 1. If you haven't got our book yet, that is exactly what it looks like. It is just called Human Biology, same name as the class by Mater and Windelspecht. I hope that's how you say it. It is the brand new edition that they just came out with, the 17th edition. So we will begin. So this chapters are separated into sections. Section 1 is called the characteristics of life. There's a few learning outcomes. This is what you should know by the end of the section. But the science of biology is the study of living organisms and the environments that they live in. All organisms have these things in common. They are organized, they acquire materials and energy, they are homeostatic, they respond to stimuli, they reproduce and have the potential for growth, and they have an evolutionary history. And we will go through each of these as we move through the section. So there are levels of biological organization. The atom is the very, very smallest level. Atoms join together to form molecules. Molecules join together to form cells. And the cell is the most basic unit of the actual living thing. So anything smaller than a cell is mostly considered the chemical compounds that make up our cells. Some organisms are single-celled. They're only made of one cell. Those are called unicellular. Some, like us, we have trillions of cells. We are called multicellular. A bunch of cells get together. They form a tissue. There's an example on the bottom right. There's one individual cardiac muscle cell. You get a bunch of those together. It forms cardiac muscle tissue. Organs are structures made up of different tissue types to perform the same function. Organ systems are made of various organs, and then organisms are made up of various organ systems. So here is our picture for that. So we have the atom and the molecule over on the left. They have a macromolecule as well. Macromolecules are the four main lipids, proteins, nucleic acids, and carbohydrates that we are all made up of. And you have the organelles, or the tiny little organs inside of cells. In the middle, there's that cell. That is the basic structure of life. Cells form tissues, tissues form organs, organs form an entire organ system, and you have a bunch of different organ systems within you, which is what this entire class is about. And we have an organism. In biology, we go higher. The organism is where it splits. That's the highest level, usually when we talk about anatomy. In biology, we also talk about our ecological levels. The organism is the smallest unit that you can talk about, but it goes into population, community, ecosystem, and biome, and then biosphere, the entire Earth. So this is just a little rundown of that. An organism is one individual living thing. You have a population of the same organisms. Community is all of the organisms, all of the living things found in that one area. Ecosystem is a little bit bigger. It's all of the living and non-living things, the soil, the sunlight, the water, the air, the oxygen is the ecosystem. And then you have a biome. We have about a dozen biomes throughout the earth. We have the desert, the tundra, the taiga, the rainforest. All of those are different biomes. They're characterized by their temperature and the plants that thrive there, actually. Like a desert has a hot temperature, and deserts are known for cacti. So that would be the desert biome. And then all the biomes put together, you have the biosphere, which is our Earth. Life requires materials and energy. Energy is just defined as the capacity to do work. Humans acquire materials and energy by eating. We are heterotrophs. We have to eat to get energy. We don't make our own, like photosynthesis, like the plants. Food provides nutrients, which are used for building blocks and energy. Metabolism is the sum of all of the chemical reactions that occur within a cell or organism. We eat our food, and there's a bunch of different chemical reactions as it travels through our digestive system. That is called our metabolism. The ultimate source of energy on life on Earth is actually the sun, because plants are at the very bottom of the food chain. They form the base of our food chains and food webs. If there are no plants, there is nothing for the animals underneath to eat. So photosynthesis, used by plants, algae, and certain bacteria, harvests energy from the sun, converts it to chemical energy, produces sugars, which serve as the basis as the food chain for other organisms. There's your basic equation of photosynthesis at the bottom. Carbon dioxide and water are taken, carbon dioxide out of the air, water out of the ground. It's converted, or it's mixed up with sunlight. Sunlight activates that process, happens in the chlorophyll of plants, and molecules get rearranged, bonds get broken and reformed. We have glucose, and then oxygen gets put back into the atmosphere, which is lucky for us. That's what we breathe. Living organisms maintain an internal environment. Homeostasis is the maintenance of a constant internal environment. Our body is constantly trying to maintain homeostasis, maintain that balance. Most organ systems strive to maintain homeostasis via a series of feedback loops. There's negative feedback loops, there's positive feedback loops. I'm going to go over that briefly, but we're going to get into each of the systems as we talk about it throughout the year. But negative feedback loops strive to maintain some kind of set point. Temperature is always the easiest one to understand, the easiest one that we use as an example. You have your set point of 98.6 degrees Fahrenheit. You get too hot, your body sweats in order to cool your body down and bring it back to that set point. If you get too cold, your body shivers, tries to make more heat in order to bring you back up to that set point. But everything, not everything, most things in our body have a set point. Our blood sugar has a set point range that we are supposed to fall in. Our heartbeat, our blood pressure, we all have a set point. And anytime our body strays from that set point, it does something to bring it back. Positive feedback loops, childbirth is the typical example we talk about. These are loops that strive to reach a goal. Nerve impulses from cervix are transmitted to the brain. Brain stimulates the pituitary gland to secrete oxytocin. Oxytocin carries body to uterus, stimulates uterine contractions because it's pushing the baby out. It's like a cheerleader. Push it out, push it out, push it out, push it out. And then once that event happens, the baby is actually born, then we go back to normal. So positive feedback loops are strive to reach some kind of a goal. Living organisms respond. Homeostasis would be impossible to maintain without the ability to respond to stimuli. Like we said, our temperature, we get too hot. That is a response to our outside environment. We sweat. Our body automatically tries to cool us down. We get too cold. Our body automatically responds. This isn't something we have to think about. We shiver to try to heat us back up. Living organisms reproduce and develop. So when organisms reproduce, they pass on genetic information to the next generation. Growth is the increase in size and number of cells, but development are all the changes that occur from fertilization until death. Every human goes through these stages, changes that occur in our childhood, adolescence, adulthood. But development also refers to the repair of cells and tissues after injury. We are constantly repairing ourselves. We're constantly shedding skin cells. Our skin always sheds. So our body from underneath has to make more. That's part of our development. Organisms have an evolutionary history. Evolution is how a population changes over time. It's important when we talk about evolution, we're always talking about individual populations. Natural selection is the mechanism or process by which evolution occurs, and the natural selection is based on adaptations, which are features or structures that help an organism survive in their environment. It is also based on variation, the variety of adaptations that occur within a population of organisms. Sometimes certain organisms within that population have a better variation that helps them survive just a little bit more, or have a few extra babies, and that is the key to survival for the whole population. When a new variation occurs that allows organisms to capture more resources, those individuals tend to have more offsprings. This is what happens a lot in antimicrobial or antibiotic resistance. You have in the GIF, there's the light pink bacteria, whatever they are, the antibiotics come along and they kill it. Well, that bacteria mutated, now it's red. And that fact allows them to resist whatever antibiotics that we are trying to throw at it. And now the pink ones are all dead, but the red ones, the mutated ones come in, they're going to reproduce because they live, and the new population that's going to survive is all going to have whatever that red gene is that allows them to resist. Next section, humans are related to other animals. So we're going to summarize the place of humans in the overall classification of living organisms. This whole branch of biology is called taxonomy, the naming of things. All life is classified into one of three domains. Bacteria, archaea, and eukarya are their names. Bacteria and archaea contain organisms called prokaryotes. These are single-celled organisms that lack a fully separated nucleus. So prokaryotic cells of various shapes. Archaeans, the domain archaea, they adapt to extreme environments. These are the bacteria you find in volcanoes and in underwater vents and in salt lakes where it's extremely salty. Bacteria are prokaryotic cells of various shapes. They can adapt to all different environments. They can photosynthesize, they can chemosynthesize, and some of them have very unique chemical characteristics. All organisms in the domain eukarya contain eukaryotic cells. These cells contain a fully separated nucleus as well as a more complex structure and organelles. Some eukaryotes are single-celled and some are multicellular, like humans. So this is just an overview of prokaryotic versus eukaryotic cells. Prokaryotes are smaller and simpler, but they have DNA in the middle. We just call that the nucleoid region. It's not fully separated by a nuclear envelope. On the right, you have the eukaryotes. You have several other organelles in there, and that's not even all of them. But its nucleus is where its DNA is found, and it's fully separated by a complete envelope. The domain eukarya is divided into 4 kingdoms and 6 supergroups. Supergroups are very, very new. We usually focus on the kingdoms: plants, fungus, animal, protists. And this is called an evolutionary tree, where the common ancestor for the very first cells are very, very, very far away from each other. And the closer you are in the tree, the more recent your ancestors branched off from each other. So our closest ancestors, according to this tree, are actually fungus. So you have the protists, algae, protozoans, slime molds, water molds. Protists are mainly known for what they're not. They're kind of a unique category and they're quite fascinating, but we don't talk about them too much in this class. The plants, kingdom Plantae, kingdom fungi, fungus, however you want to say it. And then Animalia, that's us. We're the animals. For kingdom Animalia, most animals are invertebrates. They lack the internal skeletal support structure called vertebrae. Earthworms, insects, mollusks, these are all invertebrates. And then vertebrates have the nerve cord protected by the vertebral column. Fish, reptiles, amphibians, birds. Vertebrates with hair or fur and mammary glands are called mammals. Humans are mammals, as are raccoons and seals. Humans also belong to a group called primates. These are mammals with flexible hands and feet, forward-looking eyes, and enlarged brains. All the primates descended from a common ancestor, the most recent common ancestor. Scientists have sequenced the genome of thousands and thousands of organisms. And according to the DNA sequencing, these are our closest evolutionary relatives. So it puts the chimpanzee on top. If you compare a chimpanzee up to itself, obviously the DNA is going to be 100% similar. But if you lay out all of our 3 billion base pairs next to a chimpanzee's 3 billion base pairs, we're 98.1 or 2% similar. We have a lot of similarities. So I'm going to say 98% similar. We have 2% differences in our DNA, which is very, very small amount. There's an article linked in this. I will link it in the comments. But what makes us human? That 2% of DNA, they focused on that and they found several DNA sequences and literally what makes us different. HAR1, FOXP2, AMY1, ASPN. You can read those, and each of these things are advantages that we have over our closest relatives. And then science is a process. We're going to talk about the scientific method, which is hopefully something you've talked about in every single science class you have ever taken throughout your entire life. But science is a way of knowing about the natural world. The root word sci from science literally means to know. Scientists should be objective or factual and not subjective, involve judgment, although there are some observations that we have to make the quality of, and we have to make that call. The scientific process uses the scientific method, a standard series of logical steps that scientists use across the world. Scientific method always starts with an observation. It's a formal way of watching the natural world. We gather information with our senses. Scientists like making quantitative observations involving numbers, measurements, volume, mass. We like numbers. But there are times you have to make qualitative observations where you have to describe the quality of things. So in that picture on the right, where it says qualitative data, what color would you say that is? Is it orange? Is it red? Is it red-orange? Is it orange-red? Is it red number 1, 2, 3? That's the quality of something. We can't really measure how red that square is. So sometimes you got to describe the quality of things. Then we develop a hypothesis. This is a possible explanation for the observation of a natural event. We use inductive reasoning when someone uses creative thinking to combine facts into a cohesive whole. And then scientists will make a testable prediction based on the hypothesis. Scientists will then use deductive reasoning to test the hypothesis. It will usually be an if-then logic. If I do one thing, then it's going to affect this thing in this way. They can do tests in the field, even though it's hard to control all the variables if you're doing experiments out in the field. A lot of ecological scientists try to do this. They can do it in the lab with various model organisms or even humans. Sometimes the lab won't completely reflect what's happening out in the field, though, so that's difficult. They have to balance. They can also do experiments with computer model simulations, and this has a lot to do with ethics. Well, and what you can and can't do. So scientists who want to study earthquakes can't cause an earthquake just because they want to study it. They have to either wait till an earthquake happens and then study it afterwards, or they build computer models to see what would happen. Diseases are another one. You can't give a population a disease just because you want to see what's going to happen to that population. Scientists did that one time. It's called the Tuskegee syphilis experiment, and it did not turn out well. But it is very unethical to do that. So we use computer simulations. Parts of an experiment. These are quite important. Dependent variable, independent variable, controlled variables. Dependent variables are usually the parts that are being measured. They're pretty easy to pick out, so I like to pick these out of an experiment first. Scientists have no control of these parts. They're just measuring it. What is happening when we do this? It is the effect piece of a cause and effect relationship, or the then piece of an if-then statement. The independent variable are the categories that are being compared. Scientists will manipulate these categories on purpose to be able to compare them. There's always an experimental group and a control group. Experimental groups, you can have more than one, as we'll see in a minute, and then the control group is usually, in medical terms, it's the placebo group, where you don't give them any medication and you still see how their symptoms progress. It is the cause piece of the cause and effect relationship, or the if piece, if I do this one thing. And then controlled variables, all the other conditions besides the independent variable should stay the exact same. So the effects can accurately be contributed to the independent variable. If you have all these other variables that are different, you don't know if the results are due to one of those or if they're due to the one you manipulated. So example, researchers want to know which medication reduces fever the quickest. They take 300 people with fevers, split them into 3 even groups. Their fever is measured at the start and again after 1 hour. Group 1 gets 10 cc's of ibuprofen. Group 2 gets 10 cc's of acetaminophen. Group 3 gets 10 cc's of a placebo. And then we pick out the variables. The dependent variable, the thing you are measuring, is the fever. That is a quantitative observation. You can actually measure your fever, your internal body temperature, and put a number to it. So that would be a quantitative observation. Your independent variable would be the different medications given to the 3 groups. And that is the only thing you want to differ between the 3 groups. You want everything else to be controlled variables and stay the exact same. So the amount of medication given to each group is the same. The number of people in each group is the same. The time measuring before and after 1 hour is the same. The instruments can also be the same. You're using the exact same thermometer on every single person. There's a lot of things that need to be considered and remain the same. The ideal study is called a double-blind study in medical research, where either the doctors or the patients know which group they're in. Nobody knows which medication they have. So after the experiment, you have to collect and analyze the data. Results in the form of data may be presented in a variety of formats. Graphs and tables are the most common. You've all seen tables. They look like the one on the left. And then line graphs are more commonly used, but bar graphs and pie charts, those are also used as well. Statistics are used to interpret data. Statistical data include a standard error to show how uncertain a value is. Are you 100% certain that your values are meaningful? And then statistical significance is determined to evaluate the probability that the results are due to chance or some other experimental variable, or is it due to that one independent variable that you changed that you want to see the differences in. Conclusion is next. Scientists analyze data in order to reach a conclusion about whether a hypothesis is supported or not. And in science, there's no right or wrong answer. You gather the data and your hypothesis, oh, my data supports my hypothesis or my data rejects the hypothesis. If it rejects the hypothesis, then most scientists will just go back, change the hypothesis, and start again. And then studies can be published in scientific publications, which are very prestigious. A scientific theory is an accepted explanation for how the world works. These can only be made after thousands of experiments provide overwhelming evidence. Not one scientist can make a theory by his or herself. It's a collection of thousands of experiments, evidence, data, and then the scientific community comes together as a whole and decides what is or is not a theory. Scientific challenges facing society. Science is a systematic way of acquiring knowledge about the natural world. Technology is the application of scientific knowledge to the interests of humans, because we like doing things in our best interest. And scientific investigations are the basis for the majority of our technological advances, such as the cell phone or a new drug. Diseases are what we're going to focus on. There are, of course, other challenges in this world. Climate change is a huge one, but this is a human biology class. We are trying to prepare you for microbiology and for anatomy. So diseases is a huge one. Emerging diseases are diseases that are relatively new that we have not seen before. Avian flu, the swine flu, SARS, MERS, and COVID-19 caused by the new form of SARS. Then there are re-emerging diseases known for some time and considered low risk, but for occasional outbreaks. Ebola, there's an Ebola outbreak every once in a while. The measles have started to come back because not as many people are vaccinating. Some come from new or increased exposure to certain animals. Some come from globalization. They spread more easily. The more people travel across the world, the more they will bring whatever germs they have with them. And then some come when disease vectors mutate and change hosts. The swine flu, the avian flu, and COVID all changed hosts at some point and got mutated along the way. So that is all for chapter 1. Complete the questions. Make sure you watch the video until the very, very end and complete the discussion.